So, first of all, let me also add my voice to thank and congrats the organizers, Sherry, and you guys have done wonderful work organizing this fabulous meeting. I truly enjoyed a lot about this the first session. So, can you see that in the presentation mode?
Yes. Oh, perfect. Great.
Okay. So I love to love to be here today, and thank you for the organizer for having me. And so, as you know, Ben mentioned about the one research area we are very interested in is using mRNA to restore tumor suppressor, and certainly also for protein generation. And here today, I will talk more about the cancer immunotherapy. And we are part of the Center for Nanomedicine at the Brigham Women's Hospital and Harvard Medical School. Here is our building. We are on the 7th floor, so you're probably going to see me here. I'm sitting right here. And so we all know mRNA now, it's a new medicine, and with the approval of the COVID mRNA vaccine. And this mRNA vaccine have two really important components. One is certainly mRNA itself, coding the spike protein for the virus. And the other part is the nanoparticle. Yeah, in this case, it's a lipid nanoparticle. And the use of lipid nanoparticle is truly important because mRNA itself is highly negative charge, is big, and certainly is susceptible to enzymatic degradation. And also, it's very important that we need to deliver mRNA to the cytosol of cells for protein expression. So this nanoparticle pretty much do all this work, and then to solve all the challenges for mRNA delivery. And this is why we now have these mRNA therapeutics now, vaccine now ready for infection diseases, and certainly a lot of clinical development of using mRNA for therapies. My lab actually started working with the messenger RNA about over 10 years ago, and I still remember that was like the end of the 2012. I read a report published by Boston Global talking about the company called Moderna, at that time just out of the stealth mode, and they introduced a company which is now they want to using messenger RNA to generate the proteins or restore proteins or generate new proteins. So at that time, my lab was actually working with the siRNA for gene silencing for cancer therapy. And I read the article and thought, gee, that was very, very exciting, because on one hand, my lab can use siRNA to silence the gene, but if we can adopt the mRNA, then we could restore some proteins. So literally, it's two complementary technologies that you can. With that, you can pretty much do everything you want to modulate the genes. So as I mentioned, at that time, my lab was interested in cancer therapy. So we all know cancer is highly involved with genetic mutations, and both simultaneously together with the overexpression of pro-oncogenes, as well as the loss of tumor suppressor genes. And with the loss of a balance, normal cells can become tumorigenic cells. And also this gene mutation can contribute to the tumor cell invasion, metastasis, as well as the drug resistance. So we all know, like most of pharmaceutical companies or biotech companies, they have developed personally many drugs to target the tumor suppressor drivers, like small molecules, antibodies, and other therapies. But there are really few options that are available really for the to restore the tumor suppressor gene functions. So we think that's a great opportunity really for using messenger RNA to restore the tumor suppressor. This is how we started working with messenger RNA. So there are certainly a lot of tumor suppressors, and then here I will just introduce two of them, which are probably most widely studied ones. And the first one is the P10 phosphotension homolog depleted chromosome 10. As you can see over here, there are a lot of loss of functions or mutations of these tumor suppressor gene in different tumor types. For example, in the prostate cancer cells, prostate cancer patients, in the later stage, 40 to 50% patients, they have like complete loss of the P10, and loss of P10 certainly contribute a lot of, as I mentioned, the drug resistance, and also the low response to the immune checkpoint blockade therapy. I'm gonna mention about the later on. And the other one we have been studying is the P53, which is probably most widely mutated tumor suppressor gene in many cancer types. And this is also the master regulator gene to control many biologic pathways, as as a really wonderful review article written by Dr. Scott Rowe and his colleagues. So to deliver mRNA to tumors, particularly solid tumor, is probably more challenging compared with the vaccination. You can just inject it to the intramuscular injection or subcutaneous injection, but for systemic delivery of mRNA or siRNA or other RNAs to the solid tumor is really challenging because it really involves multiple barriers. As I summarized here in this figure, you can see you need nanoparticles or other delivery strategies to protect the RNA from degradation. You need to have a relatively long circulation and good accumulation to the tumor tissues, and penetration is another challenge for big molecules to go deep to target as many tumor cells as possible. And internalization, as I mentioned, for bigger RNAs like mRNA, it's really hard. And endosomal escape and then release from the endosomes is very critical. So over the years, my lab has been developing. My lab is more like platform technology lab, so developing multiple nanoparticles. And the whole purpose for us is to really solve these other challenges for systemic delivery of mRNA or siRNA or others to the solid tumor. And so today I'm more like focus on this called solid lipid polymer hybrid nanoparticle system for tumor suppressor mRNA delivery. And so this is the platform we developed in the lab. We call it lipid polymer hybrid mRNA nanoparticle, and which is very actually similar to the lipid nanoparticle, as you can see if you look at here, the structure. And the difference is we actually add some polymers to the core of the nanoparticles, with the hope to improve the stability of the nanoparticle, as well as providing new functions to these nanoparticles. So in short, what we found is this nanoparticle can dramatically enhance the cellular uptake and endosomal escape of the for mRNA, and certainly prolong the systemic circulation and tumor accumulation. And then we showed that this nanoparticle is quite stable. Its round size is 100 nanometer, and we then with these P10 amine nanoparticles, we incubated with the P10 non-human prostate cancer cell lines, such as the PC3 cell lines in this case. And we can see after 24 hours, 48 hours, you can see very good translation, the protein expression of P10. And then the P10 restoration in the tumor cells can actually induce apoptosis by the PI3K-AKT pathway. And also we showed that dose-dependent toxicity of these P10 amine nanoparticles in, like, for example, PT3, and then also LINCAP, which is also a P10 lost prostate cancer cell lines. But meanwhile, we also tested these nanoparticles with the normal prostate epithelial cells, and then even with P10 competent prostate cancer cells. And in both cases, you can see we don't see very obvious toxicity as compared with the P10 non prostate cancer cells. So this indicates that our strategy could be potentially used for like precision medicine. For example, targeting the tumor suppressor mutated patients. So with that, we actually did some in vivo studies, and then here is the subcutaneous model of the IV injection of the nanoparticles, and we can see the inhibition of the tumor growth. And also we tested these nanoparticles in a bone model of prostate cancer. We all know, like, later stage prostate cancer has about like 80% like patients have the bone metastasis. So it's very important to target the tumor cells in bone. So that's we have the experimental model by directly inject the luciferase expressing PC3 cells to the tibia of the mouse. And you can see here with the tumor growth, you can see more luminescent signals from the IVS imaging, whole body imaging. And then with the treatment of our P10 amine nanoparticles, again, you see the inhibition of the tumor growth. So that's the story, how we get started using the P10, using mRNA to restore tumor suppressor P10. And when we were working on this project, we saw a few publications actually, for example, the first one was published in Cancer Discovery by Dr. Patrick Hu's lab when he was still in the MD Anderson. his lab found, so all these papers, regarding, the cancer patient who received the immune checkpoint blockade therapy. And in particular, in this first paper, what they found is when they compare the patients with P10 presence and the P10 absence, and then their response to the immune checkpoint blockade therapy, like a anti-PD-1 therapy, what they found is very clearly with the presence of P10, you can see the reduction of tumor volume, but with the absence of the P10, you can see the tumor growth. And also importantly, what they found is that P10 presence actually can have higher CD8 T cells infiltration in the tumor sites. And then following those papers, more recently, there's breast cancer paper, and then there's a lung cancer patient paper, the cancer discovery. All these papers show like a very consensus, like P10 loss is associated with actually immune suppressive tumor microenvironment, with a decreased T cell infiltration, with increased immune suppressive cytokines, and also poor response to the immune checkpoint blockade therapy. So we were very excited when we saw that these papers. We think, gee, that's perfect for our research. Now we have these P10 mRNA nanoparticles. Why don't we just switch from human prostate cancer cell lines to, for example, murine tumor cells? So we pick up 2 cell lines. Number one is again, it's the prostate cancer P10 cap A, which is the P10 lost tumor mouse cell line, and then the other one is the melanoma cell line B16F10, which has a P10 mutation, indicating meaning it's a loss of function of P10. So in both cases, you can see the dose-dependent toxicity, in the cells. But also very excitingly, what we saw is with the cell death, you can see the increasing of the immunogenetic cell death biomarker. This is 3 common markers: the calreticulin protein on the membrane of cells, and the release of ATP and HMGB1, extracellularly. So we think, that P10 mRNA restoration could be very exciting. Then we did a little bit of biology study, although my lab is not a biology lab. So we found like P10 restoration actually can promote the autophagy pathway based on the literature. And because P10 actually can regulate the PI3K, NKT, and mTOR pathway, and mTOR inhibition can in turn activate the autophagy pathways, and which is actually closely associated with the immune response. So when we use the inhibitor like 3-MA small molecules, you can see co-incubated with the nanoparticles, you can see the reduction of ATP release, as well as the reducing expression of the calreticulin protein on the PC3 cell, P10 kappa A cell surface. So all this indicating at least autophagy is in part contribute to the immune response. And meanwhile, if you combine this autophagy activated like rapamycin, which is we all know is mTOR inhibitor, but then activate the autophagy pathway, and the combination with the P10 mRNA nanoparticle with rapamycin gives you the even much better cell death. And so we did the in vivo studies to confirm that these make sure to evaluate whether P10 restoration can actually moderate the immune microenvironment. So we did IV injections of the P10 mRNA nanoparticles, and then we took the tumor tissues, we did the flow cytometry, look at the as you can see here, this is the affected CD8 T cells. You can see the increasing. Meanwhile, you can see the reduction of the regulatory T cells, as well as the monocytic malware-derived suppressor cells. So indicating the P10 itself can actually changing the immunosuppressive microenvironment. And then we combine these mRNA nanoparticles with PD-1 therapy. So again, here we use this melanoma cell model as a subcutaneous model, and which is doesn't respond well to PD-1 itself, as you can see here. And P10 mRNA nanoparticle itself does show some pretty good actually inhibition of tumor growth, but the combination really is the best. And then we further to confirm that the T cell response is actually from the mRNA nanoparticles, not from the anti-PD-1 itself. And then we further depleted all the T cells to show like with the T cell depletion, you can see the tumor growth can be restored in a certain level. comparable to these P10 mRNA nanoparticle itself. So again, this indicating, the CD8 T cell is very important for the anti-tumor effect. And then certainly, like to point out, the P10 mRNA itself has the anti-tumor effect, intrinsic anti-tumor effect. And so this is the P10 mRNA combination with anti-PD-1. So we think, that's exciting. So why don't we expand it to other tumor suppressors like P53, which is also, highly correlated with immune regulation in tumor tissues. So that's what we collaborated with my colleague Dan Duda from MGH, and we combined the P53 mRNA nanoparticles with the immune checkpoint blockade for hepatocellular carcinoma. His lab has the isotopic models, so we generate nanoparticles. We work very closely together, and I'd like to point it out, immune checkpoint blockade like anti-PD-1, anti-PD-L1. Recently approved for HCC treatment as a first line or second line, depending on the combination. And so, unfortunately, for HCC, still majority of patients, they don't actually respond well to the immune checkpoint blockade therapy. And we're thinking, by targeting the tumor cells, for example, correcting the P53 mutation in the HCC, which has around a third of patients have the P53 mutation, could potentially improve the immunotherapy. So that's what we did. And then we combined the P53 mRNA nanoparticles, and then with the with anti-PD-1, as you can see here, in orthotopic models, we show the combination shows the best efficacy. And very interestingly, what we thought from the tumor tissue immune microenvironment studies, P53 mRNA nanoparticle itself actually does not change much of the tumor microenvironment, which is very different from the P10 story. And only the combination of the P53 mRNA nanoparticle with PD-1, we can see the dramatic difference in terms of, like, for example, affected CD8 T cells and CD4 T cells, and then some pro-inflammatory cytokines. So this is certainly a lot of biology we have to understand, and so we are working now with Dan Duda, and then to do a lot of actually sequencing studies to understand the biologies. And then certainly we did the combination therapy to show like the combination P53 and PD-1 in a long-term studies to show like this combination is synergistic for inhibit the HCC growth in autologous models. Also, we showed like this combination can actually reduce the metastasis and other complications associated with HCC. So I think that's pretty much I'd like to share with you today. And then so come some summary of the work is I hope all data to demonstrate that mRNA nanoparticle is actually a potent strategy that can be used to restore the tumor suppressor, and such as P10, P53, and then also the reactive fission of the P10 can actually induce the immune response. And certainly another different story is a combination of P53 mRNA with immune checkpoint blockade can the combination can actually reprogram the immune microenvironment. And I don't have time to talk about other works, but we also showed that P53 can actually sensitize the tumor cells to the chemotherapies or the photodynamic therapies. And there's ongoing work, I think it's very exciting work, as that we found that tumor suppressor restoration in combination with immune moderators can actually very selectively target some certain immune suppressive cells. So hopefully we will share this work with you very soon. And with that, I'd like to thank my collaborators, Dr. Bruce Zeta from Children's Hospital here in Boston, and Dr. Dan Duda, Dr. Chuck Beveridge from University of Maryland. We work together with all these P50, P10 projects. And the team, like Arif Islam, did these initial P10 work. He's now a principal scientist and then a project leader in Sanofi mRNA Center. And Yaoxin did the P10 mRNA in combination with immunotherapy. He's a faculty. And Yulin is an instructor in the lab. She did the P53 work. And then Yudong is now working with the P10 in combination with immune moderate for specific targeting of immune suppressor cells. And also certainly would like to thank the funding support. And I think last slide to just borrow the Moderna's picture again, to show like this is an exciting area, and I think the mRNA is just the beginning for the whole field. Thank you very much, and happy to answer your questions.